Welcome. Close your eyes when you're ready and let the stillness settle over you. Far from the day, we'll drift softly into the quiet world of a tiny, timeless creature of the sea. If this brings you rest, subscribe, and there'll be a new one waiting tomorrow night. Off the coast of Liguria, where the Mediterranean runs shallow and warm over pale rock and sand, the water in late summer holds a particular quality of light. It filters down from the surface in slow, wavering columns, diffuse and golden, illuminating almost nothing and making everything it touches look slightly more still than it is. In that water, at that depth, drifting on the faint current that moves along the Italian coastline in the long afternoons of August, there is a creature so small that most people would not notice it, even if they were looking directly at it. Hold a fingernail between two fingers, the smallest fingernail on the smallest hand. The space enclosed by that nail, that narrow oval of pale keratin, is roughly the size of the entire adult animal, and within that space, if the light catches it at the right angle, something becomes visible at the center. A small, vivid coal of color, red or orange, warm and concentrated, visible through a bell of tissue so transparent it is nearly not there at all. This is Turritopsis dohrnii, a jellyfish, a hydrozoan to be precise, a member of a broad and ancient group of animals that includes corals and sea anemones, and the long-tentacled Portuguese man-of-war, though Turritopsis dohrnii shares almost nothing with any of them in terms of the thing it is capable of doing. At rest, floating in its patch of lit water off the Ligurian coast, it looks like what it is, an extremely small, extremely simple marine animal, pulsing its bell in slow, even contractions, trailing its tentacles, between 80 and 90 of them in a mature adult, in a fine, nearly invisible fringe around its perimeter. Nothing about its appearance suggests that it is remarkable. Nothing about it, held in the eye for a long moment, gives any indication at all. The transparent bell catches the light and releases it. The red center glows. The animal drifts. What it is capable of doing takes considerably longer to describe than to see, and seeing it takes patience, a specific, unhurried, attentive patience of the kind that most people do not often bring to a 4-millimeter animal in a glass tank of seawater. But the process begins at the beginning, which is where every process begins. And the beginning for Turritopsis dohrnii is a fertilized egg. The egg is nearly invisible. It develops in the water column, dividing and organizing its cells according to an ancient program, and within a short time it has become a planula, a free-swimming larva, flat and oval, covered in tiny hair-like cilia that beat rhythmically against the surrounding water and carry it forward in a slow, purposeful tumble. The planula is not looking for anything, in any sense that involves intention, but it responds to certain gradients in the water, light, chemistry, temperature, and those responses carry it, over the course of hours or days, toward a particular kind of surface, something hard, rock or shell, or the roughened substrate of a harbor wall or a pier post, something that offers, in chemical terms, a welcome signal. When the planula finds that surface, it stops swimming. It settles. The cilia that have been carrying it through open water cease their beating, and the tiny animal attaches itself to the substrate with a glue-like secretion from its anterior end. Then it begins to change. The free-swimming form retracts and reorganizes, and from the settled mass a structure begins to extend upward, a polyp rooted in place, its body a simple column of tissue with a ring of tentacles at the top that waves slowly in the water current. This is not the form most people associate with the word jellyfish. There are no bells here, No open water drifting, no luminous center. The polyp looks more like a tiny pale anemone. It feeds, it grows, it stays where it is, but the polyp is not the final form, and what happens next is the transition that produces the animal most people would recognize as a jellyfish. The polyp, in time, begins to bud. From its body, small structures emerge and detach, tiny, perfectly formed medusae, which is the technical name for the bell-shaped, free-swimming stage of a jellyfish's life. Each one rises from the polyp colony like something being released, and within days or weeks, it has grown into the adult form, the transparent bell, the trailing tentacles, the vivid red stomach at the center, drifting in the same warm, filtered light where the cycle began. This is the sexually mature stage of Turritopsis dohrnii. It is the form that produces eggs and sperm and contributes to the next generation in the ordinary way of animals. And for almost every jellyfish that has ever been described, this is also where the story ends. The medusa lives, reproduces, ages, and dies. The cycle has a direction, and the direction is forward. Except, in Turritopsis dohrnii, the direction is not fixed. When something goes wrong, and wrong in the life of a small jellyfish can mean many things, the adult medusa responds in a way that no straightforward model of animal biology would predict. Starvation is one trigger. The animal encounters an extended period without food, and something shifts. Physical damage is another, an injury to the bell or body that would in most animals simply become a wound to heal. Advanced age, the slow accumulation of cellular wear that eventually brings every living thing to the edge of its capacity, is a third. Under any of these pressures, the adult Turritopsis dohrnii begins to do something that takes a moment to fully picture. It pulls its tentacles in. One by one, or all at once, the process is gradual and quiet. The long, fine tentacles retract toward the body of the bell, drawing inward. At the same time, the bell itself begins to fold. The smooth, open curve of the adult form contracts and turns inward, the margin folding back toward the center of the animal's body. What was open becomes closed. What was extended becomes compressed. The medusa, in the process of this folding and retracting, is making itself into something much smaller than it was, something denser, more compact, less like a jellyfish, and more like a small, undifferentiated mass of tissue resting against the surface of whatever lies below it. It sinks, slowly, toward the substrate. It settles there. What it has become, at this stage, is something that biologists describe as a cyst-like mass. Not quite a polyp, not quite a medusa, a resting state, anchored to a hard surface in the dim water, externally quiet, externally showing no particular sign of what is about to happen inside it. What is about to happen inside it is one of the more unusual things that happens inside any living animal on Earth. The process is called transdifferentiation. The word itself is worth pausing on, not because the word is important, but because what it describes is genuinely strange in a quiet and specific way. Differentiation is the process by which cells become what they are, the mechanism by which a cell that begins as an undifferentiated blank acquires a particular identity and function. A muscle cell, a nerve cell, a skin cell, an organ cell. In the early stages of an embryo's development, differentiation is happening constantly. Cells are choosing, in response to chemical signals and positional information, what kind of cell they will become. And in almost every animal that has ever been studied, that choice, once made, is permanent. A muscle cell does not become a nerve cell. A skin cell does not become a muscle cell. The identity of a differentiated cell is stable and fixed, and this stability is in most biological contexts exactly what an organism requires. Transdifferentiation is the direct conversion of one differentiated cell type into another, without the intermediate step of returning to an undifferentiated state first. It does not require the cell to forget what it was and start over from a blank. It requires the cell to change its identity while remaining in some sense still a cell, still metabolically active, still physically present, still carrying out the basic functions of a living thing, even as the specific functions it is carrying out begin to shift. A muscle cell in the body of a reverting Turritopsis dohrnii transforms into a nerve cell. A skin cell transforms into a muscle cell. The conversion is direct. The transformation is real. It is the kind of thing that sounds, when described quickly like a metaphor or a simplification, the kind of phrase a writer might reach for to make something complicated feel accessible. But this is what the observations show. The mature cells of the contracted, settled medusa are reorganizing into different cell types. The animal is not dying and being replaced. It is not producing new cells from scratch. Its existing cells are becoming different cells, and those different cells are assembling themselves over the course of several days into a new configuration. Warm water, still substrate, the cyst resting in the dim light of the seafloor or the harbor wall or the rocky shelf where it settled. Nothing visible on the outside. Inside, a quiet cellular reorganization of a kind that no other animal currently known to science performs in the same way, with the same scope, under the same ordinary pressures of hunger or injury or age. The days pass in the way that days pass in shallow coastal water, slowly, with small changes in light and temperature, with the occasional movement of larger animals through the same space. The cyst does not respond to any of this. It rests, it reorganizes, and then, without announcement, something extends upward from the mass on the substrate, a polyp, small and pale, its structure simple, its tentacles beginning to reach into the surrounding water. Not the adult medusa that settled here days ago, but the juvenile form, the rooted colonial early stage form that the animal was in before it butted off its medusa copies and released them into the water column. The cycle, which moved forward, has moved backward. The medusa, under stress, has become a cyst. The cyst has reorganized and produced a polyp. From the polyp, in time, new medusa will bud and be released, and they will grow into transparent belled adults with vivid red stomachs and long trailing tentacles, and they will drift in the warm filtered light of the Mediterranean, and the whole thing will begin again. The process can repeat under the right conditions, or rather, under the wrong ones, the stressful ones. It can repeat again and again. The theoretical limit of this cycle, if the conditions that trigger reversal keep arising, has not been established. No one has demonstrated a ceiling to the number of times a single animal can complete it. The reversal, in principle, can occur indefinitely. There is a detail worth settling gently in the mind here, which is that Turritopsis dohrnii is not immortal in any simple sense. It can be eaten. It can be damaged beyond repair. It can be killed by a wide range of environmental conditions. The reversal is a biological capacity, not a guarantee, and like all biological capacities, it operates within limits set by the world the animal actually lives in. What the reversal represents is not freedom from death, but something more particular and more interesting, a documented biological mechanism by which an adult animal, under certain conditions, returns itself to an earlier developmental stage and begins again from there. This is a different thing from living a long time. It is a different thing from healing a wound. It is even a different thing from the kind of regeneration that allows some animals to regrow a lost limb or a damaged organ. Regeneration replaces what was lost. Transdifferentiation in Turritopsis dohrnii converts what exists into something it has not been since the earliest stages of the animal's life. The tissue that was the adult's bell becomes part of the juvenile polyp. The cells that were performing one function are now performing another. The animal, at the end of the process, is genuinely, biologically younger than it was at the beginning. Off the Italian coast, in the long, warm afternoons of August, the light continues to fall through the surface of the water in slow, wavering columns. It illuminates the pale rock and the sand below. It passes through the transparent bells of small animals drifting in the current. And somewhere in that water, against some surface in the dim and still zone, where light fades and the temperature holds steady, a cyst rests quietly on a substrate it settled onto some days ago. Its cells are reorganizing in the way that its cells know how to reorganize. And from the mass, something is beginning to extend upward, small, pale, tentative, reaching into the water with the specific unhurried quality of something that has all the time it needs because it has been here before, and it will arrange itself again, and then it will begin. Not because it intends to, not because it understands anything about what it is doing, but because this is what its cells know how to do, and they are doing it in the still and warm and ancient water of the Mediterranean coast, as they have done in water like this for longer than the practice of describing such things has existed. The polyp extends, the water holds it, the light, filtered and slow, settles over the whole quiet scene and stays. There is something about the scale of the animal that keeps returning when the mind tries to hold it clearly. 4 to 5 millimeters, the width of a small fingernail, a measurement so modest that the word small barely covers it. The animal exists at the edge of what the unaided human eye can resolve with any confidence. Hold a grain of rice in the mind, and then make it smaller, and then make it transparent, and then place it in moving water where the light is filtering down from above, and the surface is a long way overhead. That is approximately where Turritopsis dohrnii lives, in terms of scale, and that is the animal performing one of the most structurally unusual biological processes ever documented in a multicellular organism. The disproportion between the size of the thing and the nature of what it does is not something the animal is aware of. It has no awareness of anything in any sense that approaches the word as it is usually meant. It responds to chemical gradients and mechanical pressures and temperature changes and the presence or absence of food. It contracts and expands its bell in slow rhythmic pulses that move it at very low speeds through the water column. It extends its tentacles into the surrounding water, and when something small and edible touches them, it draws that thing toward the opening at its center and takes it in. These are not behaviors that require explanation in the sense of motive or intention. They are simply what the animal does. And when the animal is stressed, when food has been absent long enough, or when its bell has been damaged, or when the slow accumulation of cellular time has brought it to a threshold that its biology recognizes as requiring a response, it does the other thing it does. It contracts, it settles, it reorganizes, it begins again. The word begins is worth a moment. It is not a metaphor. The polyp that emerges from the reorganized cyst is genuinely an earlier developmental stage of the same organism. It is not a copy. It is not a descendant. It is not a different animal that has grown from the debris of a dying one. It is the same organism, biologically repositioned in its own life cycle, starting the forward progression again from a point it had already passed. This is the specific quality of what Turritopsis dohrnii does that places it in a category by itself. Not that it lives a long time, but that it moves through time in a direction that no standard model of animal development makes room for. The biology of development, as it has been studied and documented across the full range of animal life, runs in one direction. Cells differentiate, structures form, the organism matures, it ages, it dies. This is not a rule that anyone imposed from outside. It is simply what happens in every animal that has been carefully observed, with one small and now thoroughly documented exception drifting in the warm, shallow water of the Mediterranean coast. The exception is 4 millimeters wide. It has a transparent bell and a red stomach. It pulls its tentacles in when conditions require it, folds its bell, settles onto a hard surface, and quietly, over the course of several days, with no external indication of the process underway inside it, reorganizes its own cells into an earlier developmental form and begins again. This is what it does. This is what has been observed, documented, and published in peer-reviewed scientific literature by researchers who spent considerable time and care watching it happen. The fact of it does not require amplification or special framing. It simply is what it is, which is already a great deal. The light off the Ligurian coast moves in its slow, filtered way through the surface of the water and down through the shallow water column, and somewhere in that light, at the edge of what the eye can clearly see, the animal continues its existence. The bell pulses, the tentacles trail, the red center glows with the specific warmth of something small and alive in a large and mostly indifferent ocean. And if conditions require it, if hunger arrives, or damage, or the slow weight of accumulated cellular time, the animal will do what it does, and what emerges from the process will be, in the most literal biological sense available, a beginning. The new polyp, when it appears, extends from the same substrate where the adult once settled. Its structure is simple and small. Its tentacles reach into the surrounding water in the slow searching way of the early stage of a life that has, in a sense, started over. It will feed. It will grow. In time, it will bud new medusae from its body and release them into the water column, and they will become transparent belled adults with vivid red stomachs, drifting in the warm filtered light. The cycle does not announce its own completion. It simply continues, unhurried, unaware of itself, repeating the sequence with the same quiet biological fidelity that characterized every earlier repetition. The substrate holds the polyp, the water holds the light, and in the warm, still zone where the Mediterranean coast meets the shallow seafloor, the process that began with a fertilized egg moved through a tumbling larva and a settled colony and a free-swimming adult, and contracted back through a resting cyst to the beginning again, simply proceeds. Not for any reason that the animal could articulate, not in pursuit of any outcome the animal is capable of intending, but because this is what its cells know how to do, and the cells are doing it, as they have always done it, in the same ancient and unhurried water. The polyp extends upward from the substrate. The cycle has begun again. The water is warm, and the light is slow, and the whole quiet scene rests in the long afternoon of the Mediterranean, complete and untroubled, and already, in some small corner of it, beginning once more. At the very tip of a chromosome, packed into a space too small to see with any ordinary lens, there is a sequence of DNA that repeats itself over and over in the same short pattern, looping across the end of the strand, like a piece of text copied and pasted until it fills the available space. This sequence does not carry instructions for building a protein or signaling a cell, or doing anything that most people would recognize as genetic work. Its purpose is protective. It is a cap, a buffer, a kind of expendable margin placed at the end of something important, so that the important thing is not the first part to wear away. The comparison that appears most often in the scientific literature is to the plastic tip at the end of a shoelace. The shoelace itself, with all of its structural length and binding capacity, remains functional as long as the plastic tip holds. It is only when the tip frays and peels and finally disappears that the fraying begins to move inward into the woven material that the tip was placed there to protect. The protective tip of a chromosome is called a telomere, and like the plastic tip of a shoelace, it shortens a little every time the cell it lives inside divides. A cell divides, and the telomere loses a small portion of its length. The cell divides again, and the telomere loses a little more. This is not damage in the way that a scratch on a surface is damage. It is not an accident, not an error, not something a healthier cell would avoid. It is simply what happens when DNA is copied. The machinery that reads and reproduces the chromosome cannot quite reach all the way to the very end of the strand. Each copy is fractionally shorter than the one it was made from, and the shortening falls consistently on the telomere. The rest of the chromosome remains intact. The cap absorbs the loss over a lifetime of cell divisions in tissue after tissue throughout the body. This slow subtraction continues. The cap grows shorter with each copy. In young tissue, where division has not yet happened many times over, the telomeres are long enough that there is considerable margin remaining between their current length and the point at which the cell begins to notice the deficit. In older tissue, in cells that have been dividing and replacing themselves for decades, the telomeres are shorter, sometimes much shorter, and when they reach a length below a certain threshold, the cell responds to what it reads as a signal of structural crisis, because in nearly every other biological context, a shortened chromosome tip means damage, and damage means the cell should stop. The cell does stop. It enters a state that is neither division nor death, a kind of permanent pause, a settled stillness from which it does not emerge. Biologists call this state cellular senescence, from the Latin word for growing old. A senescent cell is alive. Its interior continues to function. It consumes energy, produces proteins, maintains its membrane, and responds to chemical signals from its surroundings. What it no longer does is divide. It has reached the end of its replicative capacity and recognized that fact, and it has chosen, if a cell can be said to choose anything, to stop rather than to copy itself, with a chromosome end too short to protect. In the early 1960s, a cell biologist named Leonard Hayflick was working with human cells in laboratory culture and noticed something that the prevailing assumption of the time would not have predicted. The assumption, held since the early 20th century experiments of Alexis Carrel, was that cells grown in culture were effectively immortal, that given the right nutrients and conditions, they would go on dividing indefinitely. Hayflick's cells were not doing that. They were dividing. Then they were dividing less. Then they were stopping and remaining stopped, no matter what nutrients were available, no matter how carefully the culture conditions were maintained. They reached a ceiling and held there. The ceiling, as Hayflick documented it across repeated experiments and multiple cell lines, sat at roughly 40 to 60 divisions for normal human cells in laboratory culture. Below that number, cells divided freely. At that number, they slowed. Beyond it, they stopped altogether and entered the permanent settled state that would later be understood as senescence. The ceiling itself carries his name now, the Hayflick limit, and what he observed in those culture dishes has held up across decades of subsequent work that normal human cells carry within them a finite count of possible divisions, and that the count is tracked with considerable precision by the length of the telomere at each chromosome's end. The particular pace of shortening varies by cell type, by tissue, by individual, and by a number of factors that research is still working to fully characterize. Some cells divide infrequently and therefore lose telomere length slowly. Neurons in certain regions of the brain divide rarely, if at all, after development is complete, and their telomere lengths reflect a different kind of constraint than that faced by the cells lining the gut, which replace themselves continuously throughout a lifetime. The gut lining turns over entirely in a matter of days, which means its cells divide far more frequently than neurons do, and their telomeres therefore shorten at a different rate. The body maintains a range of timelines, tissue by tissue, each one calibrated to the demands placed on each particular cellular population. What the telomere shortening encodes, in this sense, is not simply a countdown to death. It is something more like a record of what has already happened, a molecular log of how many times this particular cell line has been called upon to replace itself, written in units of lost repetitive sequence at the chromosome tip. A longer telomere does not mean the cell is young in any absolute sense. It means the cell has divided fewer times, and a shorter telomere does not mean the cell is about to fail. It means the cell is approaching the limit of how many times it was designed to be copied. The two things, cellular age and biological time, are related but not identical, and the telomere is the measure of only one of them. After a cell crosses into senescence, a number of things happen that extend its effects well beyond the individual cell itself. The senescent cell does not simply sit quietly in its tissue, neutral and inert. It remains metabolically active, and part of that activity involves the production and release of a particular mix of molecular signals, proteins, enzymes, and inflammatory markers that are secreted outward into the surrounding cellular environment. This mix has been characterized in the research literature under the name the senescence-associated secretory phenotype, a phrase long enough that it is almost always referred to by its abbreviation, SASP. The SASP is not a uniform or simple collection of signals. It includes dozens of individual molecular species, among them cytokines that promote inflammation, proteases that break down the structural proteins of surrounding tissue, and growth factors that can alter the behavior of neighboring cells. In the context of normal tissue maintenance, some of these signals serve useful purposes. They can recruit immune cells to a site of potential injury, or stimulate the surrounding healthy cells to divide and fill in a gap left by damage. The problem arises not from any single cell secretion, or from any single period of senescence, but from the slow accumulation over years and decades of senescent cells that are not cleared from tissue and continue to secrete this mix in a sustained and unrelenting way. The immune system is responsible, in younger tissue, for identifying and removing senescent cells before they can accumulate in significant numbers. Certain immune cells, including a subset of T cells and natural killer cells, carry the molecular recognition machinery to detect the surface markers that senescent cells express, and under normal conditions, they clear these cells with reasonable efficiency. But the immune system itself ages. One's capacity for this particular surveillance task diminishes over time. Senescent cells that would have been identified and cleared in younger tissue persist longer in older tissue, and as they persist, they secrete, and as they secrete, the chemical environment of the surrounding tissue shifts in ways that are measurable and that correlate, in the research literature, with the structural and functional changes associated with aging in organs throughout the body. This is not an isolated process affecting only a few unusual cell types. The same basic arc, telomere shortening with division, eventual senescence, SASP secretion, incomplete immune clearance, plays out in skin cells and liver cells and cells lining blood vessels and cells embedded in connective tissue and cells involved in the ongoing work of maintaining bone density and cardiovascular function. The specific timing and severity differ across tissues, and the consequences of accumulated senescence differ depending on the particular secretory profile of the senescent cells in question. But the underlying mechanism is consistent, and it is widespread, and it is gradual enough that its early stages produce no obvious signal at all. Running parallel to the slow compression of the telomere and interacting with it in ways that researchers are still working to map fully is a second process of cumulative cellular damage that does not track cell division at all. This one is ongoing, continuous, and energetically inevitable, a byproduct of the fundamental chemistry by which cells produce the power needed to do anything. The mitochondria, the structures inside cells that carry out the majority of cellular energy production, do their work through a series of chemical reactions that take electrons from fuel molecules and pass them along a chain of proteins, eventually using them to produce the energy currency that powers nearly every cellular process. The chain is efficient, but not perfectly so. At several points along it, electrons can slip away from the controlled pathway and react instead with available oxygen molecules, producing chemically reactive species, molecules carrying an unpaired electron, that is, in chemical terms, looking urgently for something to bond with. These reactive molecules are called free radicals, and what they find to bond with, when they escape the controlled environment of the energy production pathway, is whatever is nearest to them inside the cell. Whatever is nearest is often something important. The reactive species produced by mitochondrial energy work react with and alter the structure of DNA strands, producing chemical modifications that the cell's repair machinery must identify and correct. They react with proteins, changing the three-dimensional shape of molecules that depend on their precise shape to function. They react with the lipid molecules that make up cell membranes, causing a chain reaction, one altered lipid molecule producing a reactive species that alters the next, in a cascading process called lipid peroxidation that can damage a large section of membrane before the reaction runs out of available targets. This collective process, the slow accumulation of chemically mediated damage from reactive oxygen species produced during normal energy metabolism, is oxidative stress, and it has been present in living cells since mitochondria first appeared as a feature of complex cellular life. The body has defenses against it. A class of molecules called antioxidants intercepts free radicals before they reach their targets, accepting the unpaired electron and neutralizing the reactive species at the cost of their own chemical integrity. Some antioxidants are obtained through diet, compounds found in plant matter that have their own chemical capacity to absorb free radical damage without being destabilized by it. Others are produced by the cells themselves. Enzymes such as superoxide dismutase and catalase and glutathione peroxidase, which are present and active in cellular tissue throughout life, converting reactive species into harmless water and oxygen before the damage can accumulate. In younger tissue, the production of these internal antioxidant enzymes keeps reasonable pace with the rate of free radical production. The damage accrues, but the repair and neutralization mechanisms manage most of it, and the net effect on cellular function is small. Over time, the balance shifts. The production of antioxidant enzymes declines in many cell types as those cells age. The mitochondria themselves, which are copied and replaced by the cell in a process of ongoing maintenance, accumulate their own mutations in their own small separate genome, mutations that affect the efficiency and accuracy of energy production, producing more reactive byproducts per unit of useful energy than younger mitochondria would. The damage rate climbs, the repair capacity drops. The gap between them, which in young cells was narrow enough to be managed, widens into something that leaves measurable traces in the structure and function of the tissue it affects. In joints, the oxidative damage to cartilage cells contributes to the structural changes that alter their capacity to maintain the cushioning matrix between bones. In the walls of blood vessels, the damage to the cells lining the vessel interior accumulates alongside the effects of other aging processes, and the cumulative result is a gradual stiffening that alters the mechanics of blood flow. In the brain, where the cellular energy demands are particularly high and the rate of free radical production is therefore also high, the oxidative load on neuronal tissue across a lifetime is substantial. None of these are events with a single cause, and oxidative damage is never the complete explanation for any of them. It is a thread in a larger picture, consistent and present, and slow enough that its contribution is difficult to isolate cleanly, but impossible to disregard. Between telomere shortening and oxidative stress, there is a third axis along which cellular aging has been characterized, one that sits at neither the chemical nor the structural level, but somewhere in between. The genome of a cell, the full DNA sequence it carries, does not change significantly across a lifetime in most cells. The letters remain the same. What changes is which parts of the genome are read, how often, and with what consequences. These patterns of gene activity are governed by chemical marks placed on and around the DNA, tags that indicate to the cell's reading machinery which regions should be active and which should be suppressed. The system of marks and the processes that place and remove them is called epigenetics, and it is subject to change with age in ways that are measurable and consistent enough to be used in recent research as a kind of clock. The epigenetic marks associated with youthful gene activity patterns differ, in predictable and quantifiable ways, from the patterns found in older tissue. Some regions of the genome that are heavily marked for suppression in young cells gradually lose those marks with age, becoming active when they were previously quiet. Other regions lose the marks that kept them active and go quiet when they were previously working. The overall pattern drifts from the configuration of youth toward a different configuration that is not random. It is consistent enough across individuals that researchers have been able to build statistical models that predict biological age with considerable accuracy from nothing more than the epigenetic mark patterns at a selection of sites across the genome. The mark being measured in most of these models is called DNA methylation, the attachment of a small chemical group called a methyl group to certain positions along the DNA strand. Methylation at a given site tends to suppress the activity of nearby genes. Demethylation of a site tends to allow nearby genes to become more active. The pattern of which sites are methylated and which are not constitutes an epigenetic signature, and that signature shifts with age in ways that can be read and quantified. The models built from these measurements are called epigenetic clocks, and they have become one of the primary tools by which researchers measure the gap between chronological age, the simple count of years since birth, and biological age, the actual functional state of a tissue as reflected in its molecular activity patterns. What makes all of this relevant to a small jellyfish floating in the Mediterranean Sea is that when researchers began examining the biology of Turritopsis dohrnii in genomic detail. The picture they found did not look like an animal, simply running a slower version of the same programs found in its relatives. It looked like an animal running different programs altogether, in a coordinated and specific way that the research literature has found difficult to explain as anything other than an active, regulated response to the conditions of stress or aging that trigger the reversal. The genomic comparison published in 2022, in which the genome of Turritopsis dohrnii was placed alongside that of its close relative, Turritopsis rubra, a species nearly identical in size, shape, and basic biology, but without the reversal capacity, identified a set of gene families that were expressed at measurably higher levels in Turritopsis dohrnii. These were not random or scattered differences. They clustered, specifically around the same categories of cellular function that appear most consistently in aging research: DNA repair, the maintenance of telomere length, and the regulation of cell cycle progression. The genes involved in these processes were more active and differentially expressed during the reversal process in the immortal species than in its non-reversing relative. The DNA repair elevated activity means that Turritopsis dohrnii appears to be running its cellular maintenance machinery at a higher level than its relative, identifying and correcting damage more actively, and perhaps correcting it more completely. The telomere maintenance upregulation suggests that the jellyfish may be doing something at the chromosome ends during reversal that goes beyond what most cells do during ordinary replication, not simply losing telomere length with each division, as nearly all other cells and nearly all other animals do, but actively preserving or restoring it. And the cell cycle regulation differences suggest that the transitions between cell states that occur during transdifferentiation, the process by which mature cells transform directly into different cell types without returning to an intermediate stem state, are governed by a molecular control system that is in this particular animal notably more sophisticated than in the species sitting immediately beside it on the evolutionary tree. The two animals look nearly the same, placed side by side in a dish. A researcher unfamiliar with the distinction between them would need to look carefully to tell them apart. One carries a cellular program that accumulates damage, reaches its Hayflick ceiling, enters senescence, and eventually dies in the way that nearly all complex animals die. The other carries a program that responds to the conditions associated with senescence, not by continuing toward death, but by reassigning the identities of its cells, reassembling its body into an earlier form, and beginning again from a developmental stage it had already left behind. The difference between them is not in any visible feature. It is in the specific activity levels of specific gene families, in the instructions already present in the genome of each animal, and in how persistently and under what conditions those instructions are carried out. Two animals, nearly identical in every way that can be seen, carrying measurably different instructions for how long their cells are permitted to remember their own youth. The Hayflick limit is not changed by that. Telomere shortening is not eliminated by it. Oxidative damage continues to occur wherever mitochondria produce energy, which is to say, everywhere. What the jellyfish's genome appears to have arrived at, through whatever path the evolutionary history of this small hydrozoan has taken, is a coordinated set of responses to these ordinary aging processes that allows for something the research on cellular aging would not, without this animal as evidence, have suggested was achievable, not a slower version of the same arc, but a reversal of direction within it. The slow wearing of the telomere cap, the quiet accumulation of oxidative trace marks on protein and membrane, the gradual drift of epigenetic patterns away from the configurations of early development, all of these processes, in the vast majority of complex animals, move in one direction, and continue moving that way until the organism is no longer living. In this one small jellyfish, no wider than a fingernail, drifting through warm coastal water, they appear to move differently. The evidence for this is now genomic, not merely observational. It sits in the sequence comparison between two nearly identical animals, in the columns of gene expression data that separate a species capable of reversal from one that is not, and in the quiet, specific differences between those columns that researchers have only recently developed the tools to read. The chromosome ends shorten. The reactive oxygen species drift outward from their mitochondrial source. The epigenetic marks shift across a lifetime of cell divisions. These are not stories of failure or decay. They are ordinary processes running in nearly every complex living thing on the planet at their own pace and toward their own conclusion. And in the genome of two small jellyfish, nearly indistinguishable from one another, it turns out that the conclusion is not entirely the same. In the summer of 1883, somewhere along the warm, pale coastline of the Mediterranean, a collector lowered a glass vessel into shallow water and drew it back up, containing, among whatever else it held, a small hydromedusa that would not be thought about very carefully for another 113 years. The specimen was described by a German scientist named Johannes Friedrich Hildebrand and given a formal classification. It was small, a few millimeters across, transparent, unremarkable by the standards of the sea it had been taken from. Nothing in the record of that collection suggests that the animal was handled with any particular care or examined with any particular expectation. It was one specimen among many, gathered from a coastline that had been producing specimens for European natural history collections for generations. It was preserved, labeled, and placed with the other small, unremarkable things that fill the drawers of such collections, and the world moved on to other matters entirely. A glass vial in a European natural history archive is a quiet kind of place to spend a century. The ethanol holds its shape. The specimen remains suspended in it, its tiny bell unchanged, its dimensions the same as they were on the day it was taken from the water. Whatever properties it carried, whatever the living animal had been capable of, in the warm, shallow stretches of the sea where it had drifted, none of that was preserved in the vial. The vial held only the form. The capacity was gone. The question of what exactly had been missed would not be asked for a long time. In the years following that initial collection, Turritopsis dohrnii was classified and reclassified as taxonomists refined their categories for small hydrozoans, and it moved through the literature the way most minor species do, present in reference lists, occasionally collected, not the subject of sustained attention. The Mediterranean was full of small translucent jellyfish. Most of them did more or less the same things. They drifted, they fed, they reproduced in the standard fashion available to their kind. They aged and died, or were consumed. There was nothing in the literature to suggest that one among them was doing something else entirely. What eventually drew attention was not any property of the animal itself, but the ordinary curiosity of researchers who happened to observe specimens kept alive in laboratory conditions for longer than was typical. What they noticed and eventually began to document systematically was that under conditions of stress, starvation, physical damage, the accumulated wear of aging, the adult medusa did not simply deteriorate. It began instead to reverse. The bell contracted, the tentacles retracted. The animal sank toward a surface and began a process of reorganization that no one had thought to look for because no one had expected it to be there. The formal scientific documentation of this came in 1996 when a paper authored by Perrineau and colleagues was published in the journal Biological Bulletin. The paper provided the first systematic observational evidence of the complete regression from the sexually mature medusa form back to the earliest colonial polyp stage. It was careful work, as papers in that journal tend to be, methodical, precise, hedged where hedging was appropriate, and concrete where the observations supported concrete statements. What the observations supported was remarkable. An adult animal, stressed past some threshold, was able to fold itself back into an earlier form of life. Not metaphorically, not approximately. The medusa became a cyst, and the cyst became a polyp, and the polyp produced new medusae, and the cycle resumed. The paper was not ignored, but it did not immediately transform the field. The biology it described was strange enough that absorbing its implications took time, and the animal itself was difficult to study for reasons that had nothing to do with the strangeness of what it was doing and everything to do with the practicalities of keeping it alive. Turritopsis dohrnii, as a laboratory subject, is demanding in ways that are easy to underestimate before attempting to culture it. The animal is small and physically fragile. It requires live food, specifically Artemia napli, the microscopic larva of brine shrimp, and will not, in the experience of researchers who have tried, substitute dead material for living prey. The water temperature must be maintained within a narrow range, the water quality monitored and managed continuously, and the physical disturbance to the culture kept to a minimum, since the animals respond poorly to the kind of mechanical agitation that is routine in many laboratory settings. A culture that is going well can be disrupted by handling that would be unremarkable with hardier organisms. A culture that has been disrupted tends not to recover easily. The result of these practical constraints is that very few research facilities in the world have succeeded in maintaining Turritopsis dohrnii through multiple reversal cycles under controlled conditions. The knowledge required is specific, accumulated through trial and failure, and not easily transferred from a paper into practice. It is the kind of expertise that lives in the hands and notebooks of particular researchers, rather than in the methods sections of published work, and it has meant that serious, sustained observation of the animal has concentrated in the hands of a very small number of people. One of those people is Shin Kubota, a marine biologist working at Kyoto University's Seto Marine Biological Laboratory in Shirahama, Japan. The laboratory sits on a rocky stretch of the Pacific coast, a position that gives it access to the kind of clean, cold seawater that marine invertebrate culture requires, and in a temperature-controlled room within it, Kubota has maintained living cultures of Turritopsis dohrnii for more than 2 decades. The tanks are arranged in the way that working laboratory cultures tend to be arranged, not for display, not for aesthetic effect, but for access and observation, with the filtration and feeding infrastructure visible around them, the whole setup oriented toward the animals inside it, rather than toward anyone who might come to look. Kubota began working with the species after the 1996 paper, and what he found in the years of culture that followed was that the reversal documented by Pereno and colleagues was not an occasional or anomalous event. It was something the animals did reliably, under the right conditions, and something they appeared capable of doing more than once. Individual jellyfish in his cultures have been documented cycling through the reversal process more than 12 times, completing the full regression from adult medusa back to colonial polyp, and then returning again to the medusa form, repeatedly in the controlled conditions of his laboratory tanks. What that kind of sustained observation requires is a particular temperament. The work is not dramatic. The reversals, when they occur, take place over the course of several days, and the external signs of what is happening are subtle: a gradual contraction of the bell, a retraction of the tentacles, a slow sinking to the substrate. The animal does not announce what it is doing. A researcher who was not paying close attention, or who was attending to other things at the moment the process began, might miss its early stages entirely. The logbooks that Kubota has kept over his years of observation, recording water temperature, the stage of regression, the time at which each development was noted, represent a particular kind of scientific patience, the kind that produces knowledge, not through a single decisive experiment, but through the accumulation of careful attention applied consistently over a long period. There is something in the character of this work that matches the animal it is directed at. Turritopsis dohrnii is not in any obvious sense a spectacular creature. It is small and quiet, and moves through its water in the unhurried way of an animal with no particular sense of urgency about anything. The research that has illuminated what it does has had a similar quality, not a sudden reveal, not a single transformative moment, but a slow accumulation of careful observation that has gradually made a strange thing legible. And the feeding regime that Kubota's work depends on is itself a system of quiet daily labor. The Artemia nauplii that Turritopsis dohrnii requires must be hatched in parallel culture vessels and prepared fresh for each feeding. They cannot be stored in a way that preserves their viability for long. The work of maintaining a living culture of the jellyfish therefore involves the work of maintaining a living culture of its prey, and the work of maintaining that involves its own requirements for temperature, salinity, and timing. The whole system is interlocked in a way that makes any gap in attention consequential. Kubota has described the rhythm of this work in interviews over the years in terms that make it sound less like research in the conventional sense and more like a form of sustained, attentive care, the kind that is practiced not in concentrated bursts, but continuously, over years, in the quiet of an early morning or a late afternoon in a laboratory that most of the world has never heard of. What makes the work at Shirahama significant, beyond the observations it has produced, is that it has kept a living culture of this animal continuously available for scientific scrutiny, at a time when the questions that could be asked of it were becoming steadily more sophisticated. The tools available for studying the biology of cellular processes, the genomic sequencing technologies, the methods for analyzing gene expression in detail, have developed dramatically over the decades that Kubota has been maintaining his cultures, and the availability of living specimens has made it possible to apply those tools to the animal as they have become available. The most recent and detailed product of that ongoing availability is a study published in 2022 in the Proceedings of the National Academy of Sciences, in which a research team conducted a comprehensive genomic comparison between Turritopsis dohrnii and its close relative Turritopsis rubra. Turritopsis rubra is to the eye nearly identical to Turritopsis dohrnii. It occupies similar habitats, lives a similar life in most observable respects, and belongs to the same genus. The one thing it does not do is reverse its aging. When it reaches the end of its adult life, it ages and dies in the ordinary way. The two animals look very much the same. What they carry in their genomes, it turns out, is not quite the same at all. The 2022 study identified several gene families that are differentially expressed in Turritopsis dohrnii. During the reversal process, gene families associated with DNA repair, with the maintenance of telomeres at the ends of chromosomes, and with the regulation of the cell cycle. These same gene families are present in Turritopsis rubra, but in the non-immortal species, they do not show the same elevated activity that characterizes Turritopsis dohrnii during its reversal. The difference is not in the presence or absence of the genes themselves, but in how actively they are deployed when the animal is under stress, a distinction that suggests the reversal is not produced by some radical genomic novelty unique to this one species, but by the coordinated and specific upregulation of mechanisms that in other animals operate at lower levels or in more limited contexts. A printed copy of that paper with its genome comparison figures showing the sequence alignment between the two species and the regions of differential expression marked along the map is the kind of document that rewards slow reading. The figures are detailed, the regions of interest highlighted in a way that makes the differences visible without requiring the reader to hold the entire sequenced genome in mind. What the paper does, in a way that previous work could not, is give the reversal process a genomic address, a set of specific biological coordinates that describe not just that this animal does something extraordinary, but something precise about where in its biology the extraordinary thing is happening. The relationship between that paper and the original 1996 Biological Bulletin publication is a small illustration of how scientific understanding of a single organism develops over time. The 1996 paper established the fact of the reversal through careful observation. The 2022 paper began to establish the mechanism through genomic analysis. The distance between those two publications is 26 years, and across that distance the tools available for asking the question changed so completely that the 2022 paper is in some respects not answering the same question the 1996 paper raised, but a more specific and more answerable version of it, one that could only be formulated once the technology for answering it existed. That gap and what happened in it is worth staying with for a moment. In the years between 1996 and 2022, Turritopsis dohrnii existed in the scientific literature in a peculiar state, documented as real, confirmed as something that could be observed and described, but not yet explained in any mechanistic detail. The animal was known to do what it did. The biology underneath what it did remained largely opaque. For a creature whose most remarkable property was a capacity to return to an earlier form of itself, there was something apt about being present in the literature as a confirmed fact that the available methods had not yet fully caught up with. The wider public learned about Turritopsis dohrnii largely through journalism rather than through scientific literature, and the route from the laboratory in Shirahama to general awareness passed through a 2012 article in the New York Times Magazine by a writer named Nathaniel Rich. The article introduced Shun Kubota's research and the biology of the reversal process to readers who had no prior context for either, and it did so in a way that placed Kubota, his daily routines, his long years of attention to these small animals, his particular form of scientific dedication at the center of the story. The piece was followed in the years after its publication by documentary coverage from broadcasters, including the BBC, National Geographic, and Netflix, and Turritopsis dohrnii became, over the course of a decade, one of the most publicly recognized examples of extreme biology in popular science. The name of the animal, and something of what it does, made its way into the kind of general conversation that most species never reach. What popular coverage of the animal has tended to emphasize is the headline quality of the reversal, the fact that it can, in principle, be repeated indefinitely, the phrase biological immortality. What it has tended not to linger on is the gap between that headline and the measured, qualified nature of what the scientific literature actually establishes. The reversal has been documented under laboratory conditions. Its genomic correlates have been identified. The mechanisms by which individual cells change their identities during the process, the transdifferentiation through which muscle becomes nerve, skin becomes muscle, without passing through an intermediate undifferentiated state, have been described in some detail. What has not been established, because it is not the kind of thing that can be established in a laboratory, is whether the cycle continues indefinitely under the conditions the animal inhabits in the open ocean, where predation, physical damage, and environmental variability all impose constraints that controlled culture eliminates. The scientific literature on Turritopsis dohrnii is in this sense honest about the scope of what it knows. The peer-reviewed papers do not claim that the animal is immortal in any simple sense. They describe what has been observed, identify the mechanisms that appear to underlie it, and note the limits of what the current evidence supports. The more extravagant versions of the claim that this is an animal that simply does not die, that it has achieved something that defies the ordinary rules of biological life, belong to popular coverage rather than to the papers themselves. The papers are more interesting than the extravagant version, in a way, because they are more precise. They describe something real and specific, and mechanistically grounded, rather than something that sounds like a legend about a small jellyfish. Hildebrand, collecting specimens from the Mediterranean coast in the summer of 1883, had no way of knowing what he was holding. The tools for knowing it did not exist. The questions that would eventually make it knowable had not yet been formulated. The animal in his collection vessel was the same animal that Kubota has been observing in Shirahama for more than 2 decades, and the same animal that the 2022 PNAS study assigned a genomic address to, and the same animal whose image has appeared on documentary screens and in science magazine features, and in the kind of dinner table conversations that begin with someone saying they read something interesting. The century of not knowing was not a failure of attention or a gap produced by negligence. It was simply the time it took for the questions to form and for the tools to arrive that could begin answering them. That rhythm, the long initial period of not knowing, followed by the gradual development of means to know, is not particular to this species. It describes something about how knowledge of small, quiet, inconspicuous organisms tends to accumulate in general. The animals that demand attention through size or behavior or danger tend to be studied first and most thoroughly. The ones that are small and transparent and drift in warm coastal water without calling attention to themselves tend to wait. Turritopsis dohrnii waited for 113 years between its first formal description and the publication of the paper that established what it was actually doing. Given what it turned out to be doing, that is a wait of a particular kind of irony. An animal whose most significant property was a capacity to return to an earlier stage of life, going unexamined for more than a century while the world attended to other things. Kubota's logbooks, if they were ever assembled into a continuous record, would constitute one of the longer sustained observations of a single marine invertebrate species in the history of laboratory biology. The entries for any single day would be modest: water temperature, feeding time, stage of development, any signs of reversal beginning or proceeding. Taken across decades, they would amount to something unusual, a detailed record of the same small animal cycling through its extraordinary process, watched by the same patient observer in a temperature-controlled room on the Japanese coast, while the scientific understanding of what was being observed grew and deepened around it. The 1996 paper and the 2022 paper are connected by that sustained attention. The first paper established that something worth watching was happening. The second paper said something specific about what that something was. The decades between them were not empty. They contained Kubota's observations and the gradual refinement of methods and the occasional collections that provided material for new analysis. they contained, also the growing sophistication of the questions that researchers in adjacent fields were learning to ask about cellular processes, aging mechanisms, and the regulation of cell identity, questions that would eventually be brought to bear on Turritopsis dohrnii once the genomic tools made it possible. What the genomic comparison between Turritopsis dohrnii and Turritopsis rubra leaves behind in the reader, in the researcher, in anyone who spends time with the sequence alignment figures and the differential expression data, is a sense that the most interesting questions about this animal are ones that science has only recently learned how to ask. The 2022 paper is not an endpoint. It is a more precisely formulated starting position for the next set of questions. What the genes identified in that study are doing during the reversal, in molecular detail, and how exactly the coordination of their activity is triggered by the stress that initiates the process, and whether the epigenetic state of the cells during reversal corresponds to anything observable in the regulation of cellular aging in other organisms. These are questions the paper raises more than it settles. That quality of a study that resolves some things and thereby makes others more specifically askable is what good science in a developing area tends to look like from inside it. The history of Turritopsis dohrnii as a scientific subject is a history of that kind of progress, not the sudden illumination of a mystery, but the slow incremental improvement of the questions, each step of understanding making the next step visible in a way it had not been before. The glass vial from Hildebrand's 1883 collection, wherever it rests now, in whatever archive drawer, holds a form that carries no record of its own capacity. The ethanol preserves the shape, and nothing more. What was lost when the specimen was fixed is exactly the thing that took a century to understand was worth preserving. The living cycle, the capacity for reversal, the quiet cellular machinery that makes this particular small translucent animal unlike most things that share the ocean with it. The archived specimen and the living cultures in Kubota's tanks are related by 140 years of collection, observation, and gradually deepening comprehension, and the distance between them is in some sense the distance between having a thing and beginning to understand what you have. The Seto Marine Biological Laboratory, on its rocky headland above the Pacific, is not the kind of place that appears in the first few results of a general search. It is a working laboratory on a quiet coastline oriented toward the sea. It draws its water from and the organisms it has spent decades studying. The tanks of Turritopsis dohrnii within it are not dramatically lit or specially arranged. They are functional culture vessels in a temperature-controlled room, with the filtration lines and feeding equipment that serious marine invertebrate culture requires arranged around them. The animals inside drift in their filtered seawater the way they have been drifting in the various cultures maintained by the people who have worked to keep them alive, since the observations that produced the first systematic documentation of their reversal were made. The cycle the animals carry, from medusa back to cyst, from cyst to polyp, from polyp back to medusa, continues in those tanks without reference to the papers that describe it, or the researchers who observe it, or the wider human attention that has accumulated around this small species over the past 3 decades. The jellyfish in Kubota's cultures are not aware of being interesting. They are doing what they do in the water that has been prepared for them, at the temperature that sustains them, on the schedule of their own biology. The science that surrounds them is a human activity directed at them from outside. What they carry is simply their own. Two closely related animals, nearly identical in size and form and habitat, carrying measurably different instructions for how their cells respond under stress, That is what the 2022 genome comparison established, with the precision that 40 years of molecular biology made possible. Turritopsis rubra and Turritopsis dohrnii share a genus and share the sea and share most of what makes them what they are. The difference is specific and quantifiable and located in the relative activity of gene families that both animals possess. It is the kind of difference that is only visible at a level of resolution that did not exist when Hildebrand made his collection, and that required the accumulated work of everyone who maintained and observed and analyzed this species across the decades between that collection and the present moment. The most interesting questions about this animal are ones that science has only recently learned how to ask. The ones that will follow from the 2022 paper will require tools and methods that may not yet fully exist, and the patience to apply them carefully, and the sustained culture of living animals that makes application possible. In the temperature-controlled room in Shirahama, in the tanks that Kubota has been attending to for more than 2 decades, the conditions for asking those questions remain quietly in place. The animals drift in their filtered water. The cycle continues. The next question is forming, somewhere, in the manner that the best questions tend to form, slowly, out of the careful reading of what the last question produced, in the unhurried accumulation of attention that has always been the way knowledge of small, quiet creatures enters the world. Somewhere in the deep, cold dark of the North Atlantic, a large shark moves through water that has not seen sunlight in a very long time. Its movements are slow, not labored, but unhurried in the particular way of a thing that has never needed to rush. The water around it is close to freezing. The pressure at this depth is considerable, and somewhere inside the shark's eyes, held in the lens proteins that formed when the animal was young, is a faint chemical record that, when scientists finally found a way to read it, suggested this particular shark had been alive for somewhere in the region of 4 centuries. The Greenland shark Somniosus microcephalus is the longest-lived vertebrate known to science. It is also, by most measures, one of the least dramatic animals in the ocean. It grows to lengths of around 5 meters, sometimes more. Its coloring is a dull gray-brown. It moves at a speed of roughly 1 kilometer per hour on average, which is considerably slower than a person walking unhurriedly to a kitchen. It is found throughout the North Atlantic and Arctic Ocean, at depths ranging from shallow nearshore water down to more than 2,000 meters, and for most of its existence, it draws very little attention to itself. What researchers identified when they began to look carefully at populations of Greenland sharks was that this animal takes an extraordinarily long time to do anything. It grows slowly. It reaches sexual maturity slowly. The age at which a Greenland shark is first capable of reproduction is estimated at approximately 150 years. This means that most Greenland sharks alive in the ocean today were born well before the Industrial Revolution, and some of them were already old before the first accurate maps of North America were drawn. The animal that swims through that cold, dark water, filtering plankton and scavenging the occasional seal carcass, carries within its body a kind of involuntary archive. The method that allowed scientists to determine these ages is specific and worth sitting with for a moment. Lens proteins in the vertebrate eye are laid down during development, and unlike most proteins in the body, are never replaced. The crystalline core of the eye lens contains the same proteins that were formed when the animal was first growing inside its mother. These proteins contain carbon atoms, and the ratio of carbon isotopes in those atoms reflects the chemical composition of the ocean at the time the proteins were synthesized. By measuring the ratio of radiocarbon in the lens core and comparing it to known atmospheric and oceanic radiocarbon levels across time, researchers can estimate the year the animal was born. For the oldest Greenland shark sampled in a 2016 study, led by Julius Nielsen and published in Science, the estimated ages ranged up to approximately 500 years, with the most likely range for the oldest individual given as between 392 and 512 years. The central estimate was 412. 412 years. That figure does not announce itself with any particular drama when it appears on a page. It sits there in the same typeface as every other measurement in the study. But the thing it describes is a living animal moving slowly through cold water that has been alive since before Shakespeare's later plays were written, that was already old when the first sustained European settlements were being established on the eastern coast of North America, and that, if the estimate is accurate, was born into an ocean that had not yet experienced European whaling at industrial scale. The lens at the center of its eye carries the isotopic signature of that earlier ocean, unchanged, held carefully in a protein structure that does not turn over and does not degrade. Greenland sharks do not appear to have any dramatic biochemical secret that explains their longevity. Their metabolism is simply very slow, their body temperature is very low, and their pace of life across every dimension, growth, reproduction, cellular turnover, is correspondingly reduced. The cold slows everything. Damage accumulates more slowly when less is happening, and less is happening here than in almost any other vertebrate alive. It is a quieter kind of endurance than the jellyfish's reversal. There is no transformation, no reorganization of cells, no dramatic change of form. There is simply a very long, very cold, very unhurried continuation. In a laboratory tank in Mexico City, a very different kind of animal demonstrates a very different solution to the same underlying problem. The axolotl is a salamander, technically a neotenic one, meaning it retains its larval characteristics into adulthood and never undergoes the metamorphosis that other salamanders complete. It grows to around 20 to 30 centimeters in length. Its external gill plumes extend from the sides of its head in feathery arcs, and it lives entirely underwater, never leaving for land. Its coloring ranges from dark speckled gray in the wild to the pale, slightly luminous pink of the albino laboratory strain, which is the form most people encounter in photographs. What the axolotl does, that almost nothing else in the vertebrate world does quite so completely, is regenerate. If a limb is surgically removed, a fully functional replacement grows back. Not a scar, not a stump. A limb with bones, muscles, tendons, blood vessels, nerves, and skin arranged correctly and capable of normal use. The process takes weeks and involves the formation, at the wound site, of a mass of cells that lose their specialization and return to a more generalized state. This mass, the blastema, contains cells that were previously muscle, bone, skin, and connective tissue, and they divide and reorganize under molecular signals that gradually specify what each daughter cell needs to become. The result is a structure built from scratch, laid down over weeks, indistinguishable in function from the original. The axolotl regenerates more than limbs. Portions of the heart muscle, when damaged, regenerate with minimal scarring. Sections of the spinal cord, when severed in controlled experiments, reconnect and restore function. Areas of the brain, removed surgically in laboratory conditions, grow back. In each case, the mechanism involves some version of the same process: a local return to plasticity, a suspension of fixed identity in the damaged cells, followed by careful reconstruction, guided by signals from the surrounding intact tissue. What the blastema represents is a kind of local forgetting of what cells were, so they can be told what to become. The genetic and molecular work behind axolotl regeneration has been studied intensively, partly because the axolotl genome, when it was finally sequenced, turned out to be one of the largest in the animal kingdom, approximately 10 times larger than the human genome, containing what appears to be a large number of regulatory sequences that govern the switching on and off of developmental programs. Some of the same signaling pathways active during axolotl limb regeneration are also active during embryonic limb development in vertebrates generally, including in species that cannot regenerate. What the axolotl appears to have retained or maintained in a more active and accessible form is the developmental machinery that most vertebrates carry, but cannot re-engage after the embryonic period ends. The axolotl is also, in the wild, critically endangered. Its native habitat is the lake system of Xochimilco, a network of canals and shallow lakes in the southern part of Mexico City, and the pressures of urbanization, water pollution, and the introduction of non-native fish species have reduced the wild population to very low numbers. The species survives in large numbers in laboratories around the world, where it has been studied for decades and continues to be one of the most productive research animals for understanding developmental biology and regeneration. But in the canals of Mexico City, under the flat gray light of a large urban sky, the wild axolotl moves through diminished water in diminished numbers, its feathery gills fanning slowly, an animal whose capacities have been considered remarkable by science for over a century and are still not fully understood. In the tunnels beneath a research facility, through compressed substrate that approximates the underground colonies these animals have occupied for millions of years, a naked mole rat moves with the particular efficiency of an animal that has never needed to see where it is going. The naked mole rat is small, roughly the length of a large thumb, hairless, wrinkled, with large protruding incisors that extend outside the mouth and are used for digging. It is, by most visual assessments, unprepossessing. It lives in colonies of up to 300 individuals, with a strict social structure centered on a single reproductive queen. The workers, which are the majority of the colony, do not reproduce. They dig, forage, carry pups, and maintain the tunnel system. Their lives are, in most visible respects, quiet. What drew the attention of aging researchers to the naked mole rat was a set of mortality statistics that did not behave the way mammalian mortality statistics are supposed to behave. In almost every studied mammal, the probability of dying increases with age, slowly at first, then more steeply, following a curve that biologists describe using a mathematical framework established in the 19th century. In naked mole rats, studied across thousands of individuals over many years, the curve does not reliably steepen. The risk of death at age 20 in a captive naked mole rat is not measurably higher than it was at age 2. The animal's mortality rate does not show the upward slope that Gompertz, the mathematician whose work underlies the standard model, described as the signature of biological aging in mammals. Naked mole rats in captivity have lived to approximately 30 years, which is roughly 10 times longer than what body size alone would predict for a mammal. A mouse of comparable size lives 1 to 3 years. The naked mole rat does not appear to accumulate the standard cellular deterioration at the standard rate. Several mechanisms have been identified as potentially relevant. One of them involves hyaluronic acid, a compound present in the extracellular matrix of many animals. It contributes to tissue viscosity and cell signaling, but produced by naked mole rats, in a form with an unusually high molecular weight. This high molecular weight hyaluronic acid appears to trigger what cell biologists call contact inhibition, the tendency of cells to stop dividing when they make contact with neighboring cells at an earlier stage than is typical. The result is a consistent dampening of uncontrolled cell proliferation, and in practical terms, naked mole rats raised in laboratory colonies over decades have shown extremely low rates of tumor formation, far lower than comparably observed populations of other rodents. The naked mole rat queen at the colony's center is the oldest and largest animal in the group. She may be 20 years old or more, still reproductively active, moving through the tunnel system with a purposeful efficiency that does not appear to reflect the wear expected of an animal that has lived that long. The workers around her are younger, but carry out the same functions they always have, their cellular maintenance running at a rate that allows for a longevity most mammals in their size class will never approach. There is nothing dramatic about any of this from the outside. It is simply an animal that has found, over a long evolutionary history, in a resource-limited underground environment, a way of running its cellular processes with considerable care. In a small freshwater aquarium, attached to the glass wall by a basal disc no wider than the head of a nail, a single hydra extends its tentacles into the water with the slow, patient motion of an animal that has been extending tentacles since before vertebrates existed. Hydra is a genus of small freshwater cnidarians, relatives, across considerable evolutionary distance, of jellyfish and corals. A single individual is roughly a centimeter in length, a simple tube with a mouth at one end, surrounded by tentacles used for capturing tiny prey, and a basal disc at the other end for attachment. The animal is transparent enough that its internal structure is visible under a magnifying lens, and what can be seen there is a body in continuous productive motion. The body of a hydra is not stable. It replaces its entire cellular content approximately every 20 days through a population of continuously dividing stem cells located in the middle layer of the body column. Cells are produced there, migrate toward either end, differentiate into the appropriate cell type for their location, nerve cells, stinging cells, epithelial cells, perform their function for days, and then are shed. The animal maintains its form and function not through the longevity of its individual cells, but through their continuous replacement. There is no accumulation in this system of the kind that drives aging in longer-lived cells, no progressive shortening of telomeres in cells that will be shed and replaced within weeks, no gradual accumulation of oxidative damage in cells whose residence time is measured in days rather than decades. Multi-year laboratory studies of hydra populations have found no measurable increase in mortality rate over the observation periods, which have extended to years in some studies. The oldest individual hydra in these studies did not die at meaningfully higher rates than the youngest. This is not to say that hydra does not die. It does, from infection, injury, environmental stress, and the ordinary attrition of small aquarium life. But it does not appear to die more readily, simply because it is older. The risk of dying does not increase with age in the way it does in virtually every other studied animal. The molecular basis for this property appears to be connected, at least in part, to the sustained activity of a gene called FOXO. This gene encodes a transcription factor, a protein that controls whether other genes are active or quiet, and its activity has been linked to longevity and stem cell maintenance across a number of different organisms. In Hydra, FOXO remains highly active throughout adult life, sustaining the stem cell populations that drive continuous cell replacement and suppressing the kind of cellular deterioration that high turnover might otherwise introduce. In related cnidarians that do not show the same longevity properties, FOXO activity is lower and the stem cell dynamics are different. It is a small difference in what a single gene does, carried across the entire lifespan of the animal, producing a measurably different outcome. The hydra in the aquarium does not know any of this. It extends its tentacles and waits. Occasionally, a small crustacean drifts close enough, and the stinging cells fire, and the tentacles draw the prey inward. Then it waits again, attached to its glass wall in water that is perhaps a degree or two above room temperature, in a tank that receives a certain amount of diffuse light, its cells turning over at the same steady pace they have maintained since the animal settled there, maintaining the form and function that the species has maintained in essentially the same manner for a very long time indeed. Off the coast of Iceland, in cold North Atlantic water that is heavy with silt and mineral content, a clam sits in the soft sediment of the ocean floor and does very little. Arctica islandica, the ocean quahog, is a bivalve mollusk with a dark brown to black outer shell, and it is common enough in these waters that it has been commercially harvested for decades. The shells are unremarkable to look at, thick-walled, roughly oval, smoothly worn. What the shell contains, visible in a cross-section under magnification, is a record. Bivalve mollusks lay down a new growth band on their shells each year, a layer of calcium carbonate deposited during the active growing season that is slightly different in composition and color from the layer deposited during the resting season. By counting the bands in a cross-sectioned shell, the same principle that gives the age of a tree from its rings, researchers can determine how long the animal lived. For most quahog clams, the counts run to decades. The species is known to be long-lived, regularly reaching ages of over 100 years, sometimes considerably more. But in 2006, a specimen collected during a research survey of the Icelandic seabed was found, when its shell was sectioned and the bands counted, to carry more bands than any mollusk previously recorded. The count was 507. The clam had been alive since the year 1499, give or take a year. The researchers who made the count named the specimen Ming for the dynasty that was ruling China at the time of the animal's birth, and the name has remained in use in the scientific literature and in popular accounts of the discovery. Ming was, at the time of its collection, the oldest reliably documented individual animal in recorded history. It had been alive when Columbus was still sailing. It had been alive through the entirety of the age of European exploration, through the scientific revolution, through the industrial revolution, through the invention of electricity and the internal combustion engine, through 2 world wars. It spent all of that time on the floor of the North Atlantic, in cold water, in soft sediment, filtering plankton, and slowly adding layers to its shell, entirely indifferent to any of it. The longevity of Arctica islandica is not yet fully understood, but several contributing factors have been proposed. The cold water slows metabolism. Low metabolic rate means less cellular energy production, and less energy production means fewer of the reactive byproduct molecules that cause oxidative damage to cell components over time. Comparative studies have found that older quahog clams show lower rates of protein oxidative damage than older individuals of shorter-lived bivalve species, suggesting that the quahog's cellular maintenance mechanisms are operating with unusual effectiveness even in animals of very advanced age. The proteins in the tissues of an old quahog are in better condition, relative to expectation, than the proteins in comparably aged members of shorter-lived species. Something about how this animal runs its cellular economy appears to produce less damage or repair it more effectively, or both. The cross-section of Ming's shell, when it appears in published papers and documentary images, carries 507 distinct growth bands laid in a clean, legible curve from the center of the shell outward to the edge. They are not all the same width. Wider bands mark years when water temperatures were favorable and food was abundant, and the clam grew more during those seasons. Narrower bands mark harder years, years of cold and scarcity, when growth was slower. The whole record is there, pressed into calcium carbonate by the slow biological processes of an animal that had no purpose in making it, and no awareness that it was being made. A detailed, continuous, uninterrupted account of 5 centuries of North Atlantic climate, encoded in the shell of an animal that simply lived, for a very long time, in its cold patch of seafloor. There is something in the collective impression left by these animals, the shark in the dark cold water, the axolotl in its laboratory tank, the mole rat in its underground colony, the hydra on its glass wall, the clam in its soft sediment, that does not resolve into a single principle. They do not all share one secret. The Greenland shark is slow and cold. The axolotl rebuilds what it loses. The naked mole rat runs its cellular processes with unusual care and unusual materials. The hydra does not accumulate anything because everything is continuously refreshed. The quahog produces less damage and manages what it produces more effectively. These are different strategies arrived at through different evolutionary pressures, in different environments, in lineages that diverged from each other, in some cases hundreds of millions of years ago. What they share is not a mechanism. What they share is the outcome, a body that persists in some form for longer than the default setting of the biology would predict. The default setting is not fixed. Evolution has found its way around it, or through it, or past it more than once, using more than one approach in animals that look nothing alike and live nothing alike and have nothing in common except that they have managed, through one route or another, to stay alive for a very long time. The Greenland shark moves slowly through its cold, dark water, its lens carrying the record of 4 centuries, its body operating at a temperature only a few degrees above the freezing point of seawater. It does not need to rush. It has apparently never needed to rush. The time it has available for any given task is greater than the time available to almost any other vertebrate alive, and its pace reflects this, though of course the shark does not know this, and the pace preceded the knowledge by a very long margin. In the laboratory, the axolotl's forelimb extends from its body with the clean, unself-conscious functionality of a structure that was grown correctly from instructions that were always there, waiting to be used. The gill plumes move gently in the water. The regenerated limb is complete. There is no visible record of its absence, no seam, no tell, nothing to indicate that 4 weeks ago the animal was missing a structure that it now uses to navigate its tank. The biology did what it was equipped to do, and the result is ordinary, at least to the axolotl. The mole rat queen moves through the tunnel system of her colony, surrounded by workers whose cellular mortality curves decline to follow the standard slope. She has lived for years beyond what her size would predict. She will continue, in all likelihood, for years more, her reproductive activity undiminished, her tissues maintained by cellular processes that run with unusual effectiveness, her body simply continuing to run as it was designed to run for longer than almost any other mammal of her size ever does. The hydra waits on its glass wall, tentacles extended, cells turning over in the continuous cycle that has characterized the genus since long before the Cambrian diversification of animal life. It does not accumulate. It does not deteriorate in the way that accumulation causes deterioration. It is, in a functional sense, not getting older in the way that most animals get older, and it will not, unless something external ends it, simply stop on its own. And in the cold sediment of the North Atlantic floor, in water that has been this temperature and this color and this pressure for as long as this species has existed, another quahog clam adds a thin layer of calcium carbonate to the inside edge of its shell and does not register the year or the century or the slow accumulation of time passing above it and around it in a world that is almost entirely other than the one it inhabits. The band it adds tonight will look, under magnification, like all the other bands before it, a small legible line in a sequence of small legible lines, each one a year, laid down without hurry or awareness in the dark, cold, productive quiet of the seafloor. 507 bands in the shell of one clam, counted slowly in a laboratory by a researcher with patience and good light. Each one a year, each year a band, each band the same kind of evidence as the one before it, that the animal was alive and growing and doing what it had always done in cold water on a soft seafloor in the far North Atlantic. That is the whole record. That is what time looks like in certain living things when it is given a very long time to accumulate, not a drama, not a flourish, but a clean, quiet, patient line laid down in a material that holds its form in the dark water for as long as anything holds anything. On a printed page, the columns of letters sit in two long parallel rows. One column belongs to Turritopsis dohrnii. The other belongs to its close relative, a jellyfish so similar in appearance that a careful observer would need to hold them side by side under good light to tell them apart. The columns track the same regions of the genome, the same gene families, the same stretches of biological instruction, and yet, at certain points along the alignment, the two columns diverge in ways that are quiet but measurable. The genes governing DNA repair show elevated activity in one column and not the other. The genes associated with telomere maintenance are more persistently expressed. The regulatory pathways linked to stem cell function show a pattern in one species that the other simply does not carry. What the printed page holds in those two parallel columns is the difference between an animal that ages toward death and an animal that does not appear to be required to. The 2022 study that produced those columns was published in the Proceedings of the National Academy of Sciences. And what made its figures particularly interesting to the researchers who read them was not only what the jellyfish did differently from its relative, but what it did similarly to something else entirely. Among the gene families showing elevated expression during the reversal process in Turritopsis dohrnii were families also involved in a pathway developed in a mammalian laboratory on the other side of the world by researchers working on cells that had nothing to do with jellyfish at all. The overlap was not complete, and the researchers were careful about the language they used to describe it. But it was there, in the data, legible in the same way that the red stomach of a 4mm jellyfish is legible through its transparent bell, small, clear, and not easily dismissed. In 2006, a Japanese scientist named Shinya Yamanaka demonstrated that a mature mammalian cell, a skin cell, a liver cell, a fully differentiated cell that had long since settled into its final functional identity, could be returned to a stem-like state by introducing 4 specific transcription factors into its nucleus. The process came to be called induced pluripotency, and the reprogrammed cells came to be called induced pluripotent stem cells, or iPSCs. What Yamanaka's work showed, in a way that surprised a great many people who followed cell biology closely, was that the identity a cell had assumed over years of development was not as fixed as it had appeared. The molecular instructions that said this is what you are and this is all you will be could apparently be overwritten, not easily and not without consequences that required careful management, but overwritten nonetheless. What the genomic comparison in the 2022 paper suggested, carefully and without overclaiming, was that a jellyfish in the Mediterranean Sea had been doing something conceptually adjacent to Yamanaka's protocol for a very long time. Not identically, not through the same exact molecular actors, but along overlapping lines, through gene families that participate in similar cellular processes in a way that made the parallel worth noting and worth following. The jellyfish had not read the literature. It had simply arrived, through whatever evolutionary pressures shaped its lineage, at a cellular strategy that human researchers were only beginning to describe in laboratory terms. The data on the printed page did not prove the two processes were the same. It suggested that the problem they each solved, how to move a cell backward through developmental time, had more than one road leading to it, and that at least two very different forms of life had found roads that ran for part of their length in the same direction. Moving from that printed page to the living animal it describes requires a certain deliberate slowing. The sequence data is dense and precise and exists on screens and in journals and in the minds of researchers who have spent years learning how to read it. The jellyfish exists in warm, shallow coastal water, pulsing slowly, entirely indifferent to what the sequence data says about it. The distance between those two things is real and worth holding for a moment before moving forward. Shin Kubota's laboratory at the Seto Marine Biological Laboratory in Shirahama, Japan, is one of the places where that distance is smallest. The laboratory sits on a rocky coastal headland above the Pacific, and inside it, in a temperature-controlled room fed by filtered seawater lines, tanks of live Turritopsis dohrnii have been running for more than 2 decades. Kubota has observed individual jellyfish cycling through the reversal process more than a dozen times under his direct care. He records the time, the water temperature, the stage of regression in a physical logbook, the kind of record that accumulates slowly and is worth a great deal precisely because of how long it has been kept. The jellyfish in those tanks are not performing for the record. They are simply continuing the cycle that the genome makes possible, in the same way they would in open water, in the same way they have for as long as the species has existed. Maintaining them is not simple work. And Turritopsis dohrnii requires live food, specifically Artemia naupli, the microscopic larva of brine shrimp, which the jellyfish will not substitute with dead material. The water temperature must be managed carefully. The cultures must be disturbed as little as possible. Very few research facilities worldwide have succeeded in keeping the species alive through multiple reversal cycles, not because the biology is uncooperative, but because the animal's requirements are precise and uncompromising, and meeting them consistently across months and years is the kind of labor that does not accommodate shortcuts. Kubota has done it longer than almost anyone else. The logbook reflects that. The tanks reflect that. The jellyfish in the tanks drifting slowly in their filtered seawater reflect it in the only way they are capable of, by continuing. The question the genomic data and the logbook together raise, quietly, without urgency, is what it would mean to apply what Turritopsis dohrnii does to cells that do not belong to jellyfish. The question is not new. and several different lines of research have been following it from different directions, with different tools, for a number of years now. One of those lines concerns the senescent cells that accumulate in aging tissues. These are cells that have stopped dividing but have not been cleared by the body, and they persist in tissues releasing a chemical mixture of inflammatory proteins and enzymes that gradually degrades the function of the healthy cells around them. The mixture is precise enough to have a name, the senescence-associated secretory phenotype, and its effects have been documented in enough detail that researchers have been working for some years on compounds designed specifically to remove the cells producing it. The compounds are called senolytics, and the combination of dasatinib and quercetin has been among the most studied pairings in early clinical trials. The goal of senolytic therapy is not to slow the accumulation of damage in aging tissue. The goal is to reverse a specific feature of that damage by eliminating the cells responsible for it, to restore, in some meaningful measure, the conditions of tissue that has not yet accumulated as many of those cells. The mechanism is different from what Turritopsis dohrnii does. The cellular actors involved are not the same, but the foundational orientation toward restoration, rather than merely toward slowing, runs along a similar line of reasoning, and researchers working on senolytics are aware of that parallel, even when they are careful not to overstate it. There is another line of research that runs closer still to what the jellyfish does. Steve Horvath, a biostatistician at UCLA, developed a method for measuring biological age through patterns of chemical modification across hundreds of specific sites in the genome. The modifications are called DNA methylation marks, and they accumulate and shift in consistent patterns as an organism ages, so consistently that the pattern can be read as a clock, a way of determining, from a tissue sample, not how old an organism is in calendar years, but how old its cells appear to be by biological measure. The clock does not correspond exactly to chronological age in every individual, and the discrepancy is itself informative. A person whose biological age reads younger than their calendar age, by Horvath's measure, tends to show better health outcomes on several related metrics. Researchers comparing the medusa form of Turritopsis dohrnii to the reverted polyp form have observed something that the epigenetic clock framework makes legible in a particular way. The two forms show measurable differences in epigenetic regulation, in the pattern of those methylation marks across the genome. What that difference suggests, held carefully within what the data actually says, is that the animal may not merely be reorganizing its cells during the reversal process. It may be actively shifting the epigenetic signature of those cells towards something that reads as younger by the same kind of measure Horvath's clock uses. The animal may be, in some operational sense, rewriting its own biological age as part of the reversal. The data does not prove this conclusively, and the researchers who work in this area are clear about the limits of the evidence, but the possibility it opens is one that the field has been thinking about carefully, because what it would mean, if it held under further investigation, is that the age signature of a cell is not simply a record of elapsed time, but something more like a writable state, one that certain biological processes can alter in a direction opposite to the one in which time usually drives it. That possibility is part of what has attracted substantial investment to cellular rejuvenation research in recent years. Companies including Altos Labs and Unity Biotechnology have directed hundreds of millions of dollars toward programs conceptually informed by organisms like Turritopsis dohrnii, and by the broader question of whether cellular aging is a process that can be reversed rather than merely slowed. The specific challenge those programs are working on is one of precision, how to achieve a reprogramming effect that restores cellular youthfulness without triggering the kind of uncontrolled cell proliferation that produces tumors. Yamanaka's original protocol, applied continuously and without modification, produces cells that are no longer constrained by their developmental identity in ways that are not entirely safe. What researchers at Altos Labs and elsewhere have been exploring is partial reprogramming, brief controlled exposures to the reprogramming signals that shift cells toward a more youthful state without fully erasing the identity they need to perform their function in tissue. The practical difficulty of this work is considerable, and the timeline for any clinical application is long and uncertain. This is where the work is, not where it has arrived. The jellyfish in Kubota's tanks does not provide a blueprint that can be transferred directly from marine invertebrate to mammalian tissue. The biology is different in ways that matter. What it provides, instead, is a proof of concept that is genuinely useful, the demonstration that a living organism can execute a coordinated cellular reversal, not a slowing of aging, not a pause, but a reversal of it without destroying the organism in the process. That demonstration has been sitting in the peer-reviewed literature since 1996, and the tools needed to begin understanding how it works at the molecular level have only become available in the years since. The research is catching up to the biology, incrementally and carefully, in the way that research does. Before the work was caught up to by research, it was caught up to by writing. In 2012, a journalist named Nathaniel Rich published a long article in the New York Times Magazine about Shin Kubota and his jellyfish tanks and the biology of cellular reversal. The article introduced a wide general audience to what the species does and to the patient, methodical character of the work Kubota had been doing for years. It was followed by documentary coverage on several major platforms, and the jellyfish became one of the more publicly recognized examples of extreme biology in popular science communication, not because it was the rarest living thing or the most difficult to study, but because what it does is genuinely and simply remarkable, and because the language for describing it is accessible in a way that the language of genomics is not always. A 4-millimeter jellyfish that can reverse its own aging is a thing a person can hold in the mind without training. The data columns and the methylation marks and the transcription factor protocols require context to make sense of, and that context takes time to build. The jellyfish requires only a moment. It drifts in warm water, it has a transparent bell, and a red stomach. When conditions become difficult enough, it retreats to a surface and begins to reorganize itself from the inside outward, and when it has finished, it begins again. That is the fact, and the fact is sufficient on its own terms, even before the genomics is layered over it. What the public attention to Turritopsis dohrnii has done, beyond generating interest in the species itself, is open a door into a broader conversation about what aging is, and whether the way most organisms experience it is the only way it can go. That conversation was already happening in research contexts before the jellyfish became publicly known. The biology of aging has been a serious scientific question for decades, approached from many directions by many different research communities. But the jellyfish provides a particular kind of anchor for that conversation because it is a living counterexample, not a theoretical one, not a model system in a dish, a living animal in the ocean right now that does not follow the rules that most animals follow about the direction time moves through their cells. The rules it follows instead are ones science is still in the process of reading. The 2022 genomic study was not the last word on the subject. It was a careful and informative step in an ongoing process of inquiry that will continue to add detail and nuance to the picture over the years ahead. Other studies will refine the gene expression data. Better tools for reading epigenetic age signatures will make the comparison between medusa and polyp forms more precise. The connection to Yamanaka's reprogramming factors will be explored in more controlled experimental systems to determine how deep the parallel actually runs. The work will continue, slowly and incrementally, in the way that science does when the question is genuinely difficult and genuinely worth answering. In a research facility where the work of partial reprogramming is being carried out, a dish of mammalian cells sits under a fluorescent microscope. The cells have been treated with a modified protocol, a brief exposure to a subset of the Yamanaka factors, calibrated to shift the cells toward a more youthful functional state without fully erasing their identity. Under the microscope, against the dark field of the imaging chamber, the cells begin to show markers that indicate something has shifted in their internal regulation. The markers are specific proteins expressed at levels that correlate with younger cell function, visible as faint fluorescent signals in the imaging system. A researcher records what the microscope shows. The observation is added to a dataset. The dataset is added to an ongoing analysis. The analysis will eventually contribute to a paper, and the paper will be read by other researchers who will design the next experiment. The process is slow and careful and moves one step at a time, and it has been moving that way for long enough that the accumulated steps now constitute a substantial distance from where it started. Somewhere in that accumulated distance is a conceptual line that runs back through the genomic comparison in the PNAS paper, through Kubota's logbook, through the 1996 Biological Bulletin paper, through a century of unremarkable classification, to a glass collection vessel lifted from warm Mediterranean water in 1883, containing a small jellyfish that had not yet been recognized as anything unusual. The line is not straight, and it passes through many different kinds of work done by many different people in many different places, but it connects, and following it backward from the fluorescent markers in the microscope dish to the jellyfish in the collection vessel is an exercise that makes the distance traveled feel both large and entirely continuous, one thing leading to another, each step following from what the previous step found, across more than a century of patient attention paid to a 4-millimeter animal in shallow coastal water. The attention is ongoing. Kubota's tanks are still running. The filtered seawater lines are still supplying water at the right temperature. The Artemia nauplii are still being cultured in parallel vessels alongside the jellyfish. The logbook is still receiving entries. Somewhere in those tanks, individual jellyfish are at various points in their cycle, some feeding, some pulsing through open water, some perhaps beginning the slow retraction of tentacles that indicates a reversal is underway. The cycle does not require observers to proceed. It has been proceeding in open water and in carefully maintained laboratory conditions for as long as the species has existed. The observers have simply made it visible in ways it was not visible before. A clinical room in a different kind of facility holds an early trial participant seated while a senolytic compound is administered. The mechanism at work in the joint being treated involves the targeted removal of cells that have accumulated there over years and stopped contributing to the tissue's function, while continuing to signal the surrounding tissue in ways that degrade it. The mechanism does not resemble what a jellyfish does when it folds its bell and sinks toward the seafloor. The molecular actors are different. The cellular context is different. The organism is different in almost every way that matters, biologically, and yet the orientation is similar. The removal of what is accumulated, the restoration of conditions that were present before the accumulation began, the direction of the intervention running opposite to the direction in which time usually moves through tissue. The line between the clinical room and the Mediterranean coast is long and indirect and passes through many different kinds of science. But it exists. What makes the jellyfish useful as an anchor for all of this is not that it provides answers. It does not provide answers. It provides a standing demonstration that certain questions, which might otherwise seem to assume their own impossibility, do not actually contain that assumption. The question of whether a mature organism can reverse the cellular signatures of its own aging does not assume its own impossibility, because an organism that weighs a fraction of a gram and drifts in warm, shallow water has been answering that question affirmatively in open water without any laboratory assistance for longer than science has been asking it in formal terms. That is a quiet fact and a large one. It does not need to be held at high volume to be felt. The jellyfish is not a solution to aging and was never proposed as one. It is a data point, the most unusual data point in a field full of unusual data points that tells researchers something true about the range of what biology permits. The range is wider than it appeared before the data point existed. That widening is real and documented, and is the kind of thing that changes the direction of inquiry without changing the pace at which inquiry proceeds. Careful work is still careful work. The jellyfish drifts in warm water and continues the cycle, and the researchers at their laboratory benches continue following the questions the cycle raises, and the two activities are connected by a thread of attention that is long and patient and shows no sign of breaking. In Kubota's temperature-controlled room, the light is low over the tanks. The seawater inside them is slightly warmer than the Pacific outside the laboratory walls, calibrated to the conditions the species prefers. The jellyfish in the tanks move in the way that jellyfish move, the slow rhythmic contraction and relaxation of the bell, the tentacles trailing in the water, the red center visible through the transparent membrane that surrounds it. They are not aware of the laboratory. They are not aware of the logbook or the genomic studies or the research programs funded with hundreds of millions of dollars at facilities on the other side of the world. They are aware, if that is the right word, of the temperature of the water and the presence of food and the particular internal state of their own cells, which will eventually, when conditions require it, initiate the cellular program that allows the cycle to begin again. The cycle has been running, by the estimate of researchers who study the species, for a very long time. How long is not precisely established. The species has been documented since 1883 and has certainly been present in the Mediterranean for much longer than that. Whether individual lineages of the animal have been cycling continuously for centuries or longer is a question the current evidence does not answer. What the evidence does establish is that the mechanism for the cycle is encoded in the genome, that the genome carries the necessary instructions in a form that can be read and compared to related species, and that the comparison reveals a coordinated set of genetic differences that distinguish an animal capable of this from one that is not. The differences are not simple. They involve multiple gene families, multiple regulatory pathways, and a coordination between them that the 2022 study described as a reprogramming response rather than a single, isolated mechanism. The jellyfish does not do one unusual thing. It does many things in a particular coordinated sequence, and the sequence produces an outcome that no other animal is documented to produce in the same way. A researcher reading the 2022 paper for the first time, following the sequence alignment figures and the gene expression data across its pages, encounters a picture that is both precise and open, precise in what the data shows, and open in what the data does not yet explain. The precise parts concern which genes are differentially expressed and in which direction. The open parts concern what those genes are actually doing during the reversal process, how the coordination between them is regulated, what triggers the reprogramming response to initiate, and how the resulting cellular state is maintained long enough to reorganize the entire organism into a new developmental form. Those open parts are the current edge of the question, and they are where the work is being done now, slowly and carefully, in the way that the most interesting scientific questions tend to be worked on, not in a rush, but with sustained and patient attention. The sustained attention is itself a kind of parallel to the animal being studied. Kubota has given more than 2 decades to his tanks of live jellyfish. The species in those tanks and in open water has been going about its cycle for far longer than that. The attention and the cycle coexist without either one being aware of the other, and what the attention has produced, the logbook, the peer-reviewed papers, the genomic comparisons, the conceptual lines leading to mammalian cell research, accumulated in the same slow way that the cycle itself accumulates. One observation at a time, one division at a time, one careful record added to the previous one. The researchers who will publish the next paper about Turritopsis dohrnii are at work now in laboratories in Japan and Spain and other countries where the species has been studied. Some of them are culturing live specimens, some are analyzing sequence data, some are designing experiments to test specific hypotheses about the regulatory mechanisms of the reversal process. None of them are certain where the work will lead, because certainty of that kind is not available before the experiments have been run and the data has been gathered and the analysis has been done. What they are certain of is that the animal exists, that it does what has been documented to do, and that the documentation so far raises more questions than it answers. That is a common condition in active research, and it is not a condition of frustration. It is a condition of genuine inquiry, in which the questions are clearly formed enough to be tested and interesting enough to be worth testing. The Mediterranean coast holds many things in the water near its surface. Currents moving through the Strait of Gibraltar carry Atlantic water into the basin, where it warms and circulates in patterns that have been documented by oceanographers for decades. Nutrient upwellings from deeper water support the planktonic communities on which many small marine animals depend, including the tiny prey organisms that support a 4-millimeter jellyfish, whose transparent bell catches the filtered. The jellyfish is not the most visible creature in that water. It is among the smallest. From the surface, looking down through clear water in good light, a person lying on a boat or standing at the edge of a dock would not easily see one. It would require a particular quality of attention, close, slow, patient, to make out the translucent bell moving through the water column, the red stomach at its center. That quality of attention, applied to a very small animal in warm, shallow water, produced the question. The question, carried through laboratories and logbooks and genomic sequencing platforms and peer-reviewed journals, produced a different kind of attention applied to data columns on printed pages and to cells in dishes under fluorescence microscopes. The two kinds of attention are not the same, but they share a direction. Both are oriented toward the same small animal and what it does. Both accumulate their observations incrementally, without rushing toward conclusions the evidence has not yet supported. Both are ongoing. Outside the laboratory in Shirahama, the Pacific moves in long swells against the rocky headland. The water is cold there, colder than the Mediterranean water where the species was first described. Inside the laboratory, the tanks are warm and still, and lit by the low aquarium light that the jellyfish require